It's good?
Yeah.
Okay. So today we'll talk about the natural history of the DNA molecule. And this year we celebrated 70 years that you know the structure of the DNA. And this is a fascinating molecule. But we know a little about the orange of the DNA. And I'll try explain a little bit how I see this history. So, but it's also fascinating when you think that the genetic developing during 14 years without know how was the hereditary molecule. The first evidence about this was present in 1944 in a very interesting experiment conducted by Avery and McLeod in McCartan. In reality, they reanalyze the Griffiths experiment, but in this new version of this experiment, they're introducing a new step. And this step was very important to conclude that the DNA was the hereditary material in the biological system, because they degraded one by one the major biological molecules, and only when they degraded the DNA, the power of transformation, the biological compounds lost the power to transformation of the bacteria. This was the first evidence that the DNA was the material genetic. But the other very important experiment was conducted by Alfred Heschler and Martha Chase in 1952, when they observed that during virus infection, the DNA was introduced in the cell, and this molecule was that had the capacity to produce new viral particles. But the final evidence that the DNA was the genetic material was present in 1953 in a classic paper wrote by Francis Crick and James Watt. Evidently, it's impossible to talk about this data and forgot Rosalind Franklin. Rosalind Franklin had the best photograph about the DNA in that time. So this experimental, in reality, is a theoretical paper, inaugurated a new era in the biology that you call molecular biology. In that moment, the DNA was the most important molecule in the living beings. Okay, but at the same time, was formed a group of research that you call RNA type club, because the next question was how the information passed from the DNA to the protein. This is a very important, because this is if you understand this, you understand how the information, how is the flux information in the biological system. The first suggestion about how this information passed from DNA to protein was present by Francis Crick in a very important paper wrote in 1956. This paper is very interesting because this manuscript never was published. This manuscript only was shared by the members of the RNA type club. In this paper, it's the first time that we had a proposition about the flux information in the biological system. Crick proposed in this paper that the DNA can self-replication, can do self-replication, the RNA can self-replication, can do self-replication. They suggested, Crick suggested in this paper that the DNA can pass the information to the protein directly, but they also suggest that the DNA pass the information to an intermediary molecule that in this case is the RNA, and the RNA pass this information to the protein. And also, Crick also proposed that the RNA can produce a DNA molecule. But in the same page, the Crick say, but never the information can pass from proteins to the nucleic acids. This is the first time that you can start to understand how the information pass in the biological system. In that moment, the DNA was the most important molecule in the living beings. But in the next years, the comprehension about the molecular function in the biological system put other molecule at the center of the discussion as the most important molecule in the biological system. And three independent papers were published, one in 1969 by Calvus, and two other papers in 17, published by Larry Argel and Francis Crick, proposed that the RNA was the first molecule in the biological system. At the same year, was discovered a molecule that had the capacity to synthesize a molecule, a DNA molecule from an RNA molecule. This enzyme you call heterotranscriptase or reverse transcriptase. This was a very important point because in this moment, the RNA start to be considered the most important molecule in evolutionary terms to the biological system. And in 1984, and 82, and 1884, two independent articles were published, one conducted by Thomas Check and the other by Sidney Altman. And in those papers was shown that the RNA had other characteristics. In this case, the characteristic that was described in those papers was it was the RNA had the characteristic, the capacity to execute catalysis. This is the first time. So in that moment, the RNA had the function of the pass the information from DNA to protein. And the RNA composes the ribosome. The RNA composes the adaptive molecule, the tRNA. And now the RNA had the capacity of catalysis. Oh, so the RNA had the capacity to do all basic functions in the biological system. So this group of evidence make that in 1986, Walter Gilbert proposed the RNA world. That is the most popular model to origin of life. In this model, the RNA, only RNA organized the primitive biological system, store the information, and do catalytic reactions. So here we have a flux of the information when you look for the natural history of the living beings. The first molecule, informational molecule, was the RNA, and that passed to a stage when you had RNA and protein. And the last stage, we had DNA that passed the information to RNA, and the RNA passed the information to protein. And how can you see in this picture, all of these events occurred before the emergence of the last common ancestor. Okay, the last common ancestor are here, and all these transitions are in a step before the emergence of cells. So when you look in the history of life, we had different moments when you can think different way to pass the information. And at the first moment, we had RNA that by self-replication created other RNA molecule. This RNA molecule can create other RNA molecule, and the replication, maintaining the constant replications and constant amount of the RNA molecules in the system. And in this model, the RNA also do the catalysis. The second stage, the first transition is to pass this model to the next model, when in the ribonucleoprotein world occurred the emergence of the translation system. But in this case, the translation system is organized also by RNA. So we had RNA that can replicate it, and other molecules of RNA can translate this information stored in the RNA and pass this information for a new world, the world of proteins. In this moment, the catalytic reactions change, and now the protein assume these positions like we can see in the modern organized. And the last step in this flux of information, the natural history of the flux information, was pass the information stored in the RNA to the DNA. That is the way that you know in the modern organized. Okay, but to understand these major transitions, we need to look to the past. Today, when I look for the biological organization, basal biological organization, and today we can see that we have two basal lineages that's composed by Archaea and bacteria, in a derivative lineage composed by Eukarya. So if you want to understand the emergence of the DNA, I think we need to look for the emergence of the bacteria and Archaea. In this model here, we still had the idea of the bacteria and Archaea had one unique origin, that unique common ancestor, in this case, a cellular common ancestor, right? But we had a lot of evidence that the most simple scenario to emergence of this two basal lineage is that the ancestral of the archaea and bacteria is not uncellular organism. So the question that you need to do now is, did the cell have only one origin? What supporting the idea that the cell had only one origin? I can think a lot of the answers to this question, but here I just will talk about two answers. The first answer is the origin of the membrane. And yesterday, Francis Prodos told a little bit about this. When you're talking about the membrane, in general, we think that the membrane is similar. But when you look in details, the membranes of archaea and bacteria, we can see that these membranes are so different in constitution. Here you can see typical lipid of archaea membrane, and here in bacteria and in eukaryotic membrane. We have here L-glycerol, here we have D-glycerol. The leakage is ester here, and here in archaea is ether. The phosphate are binding in different positions. And when you look at the layers of the membrane, in bacteria we have bilayered, and in archaea in general, in some group we have a monolayer. And we had evidently we have other difference between archaea and bacteria, but it's the most basal difference between them. Other important data that when you look for the biosynthetic pathways of these lipids of membrane, archaea and bacteria had a completely different pathways. And it's very interesting because the archaea pathway we just can found in archaea. In the bacteria, we just can found in bacteria. Evidently, there was some event of lateral gene transfer, but we can identify these events. So this is the first suggestion that the process of compartmentalization occurred twice in the origin of the basal lineages. So, okay, and about the origin of the DNA, what we know? Back to this picture, is practically in all scientific community are agreeing that the RNA was the first genetic material. So if you want to understand how emerged the DNA, we need to start to understand how emerged the RNA and how this information passed to the DNA. In 2021, we published this paper. You can see this picture here. This picture is on tree and structural tree to different kinds of polymerases. So here on the top we have the RNA-dependent RNA polymerases. These polymerases replicated RNA molecules, and probably it was the first polymerase to emerge in the history of life. And closed of this RNA polymerase, we can find the reverse transcriptase, and most derivated we can find DNA polymerases. So it's very interesting because you can identify in this picture the same process that we can identify in this other picture. Okay, so in 2017, we published this paper when we proposed the tRNA core hypothesis. And in this hypothesis, we suggested that the tRNAs, or proto-tRNAs, orchestrated the initial organization of the biological system. And in this model, we proposed that the tRNAs gave origin to the PTC. PTC is the catalytic part of the ribosome, the large subunit. The PTC is the part that put together one amino acid and other amino acids and by a peptidic bond. And we also proposed that the tRNAs gave origin to the proto-mRNAs. With this was formed a proto-translation system and start the production of the protoproteins, and these protoproteins initially back to the system to stabilize these RNA molecules. There's different RNA molecules. So I would like to remember that in the modern organized, the tRNA is not translated in protein. Okay, so what you did, we constructed a database with all tRNA sequences available in the main database, and from these tRNA sequences that we get from modern organized, we have constructed the ancestral sequence for each one kind of tRNAs and translated the information stored in the proto-tRNAs, the ancestral molecules of tRNAs. And for our surprise, when I translated this information, I found similarities with the RNA-dependent RNA polymerases. That is the polymerase. This is the polymerase that replicated RNA genomes. So in the modern organized, you can find this enzyme. This enzyme we can find in virus, in RNA virus, but in cellular organized, we also can find. But in the cellular organized, the enzymes are involved in the amplification of the no-coding RNA that are very important in the regulation of gene expression. So this paper was published in 2017 in Frontiers in Genetics, and here you can see the structure of the RNA-dependent RNA polymerases derivated from the translation of the tRNA ancestral sequence. Okay, this is a small part. This is very interesting because when you look for when you compare this part of the protein that you can find a similarity with the RNA-dependent RNA polymerase, this part is similar to the catalytic part of the modern RNA polymerase, RNA-dependent RNA polymerases. So in that moment, we are using two different approaches. The first approach we called bottom-up. In the bottom-up, we translated the tRNA ancestral sequence. So we are in this movement around the tree of life, and we also used a top-down approach. In this approach, we constructed other databases, but in that case, only with the RNA-dependent RNA polymerase from modern organisms and reconstructed the ancestral sequence from this enzyme. So if my methodology was correct or is good, in a moment of three, two methodology will match in determinate point here. So we did other kind of analysis. We analyzed the capacity of binding that these small peptides here, and we use different algorithms to predict the ligands of the different ancestral sequence to the RNA-dependent RNA polymerase. It's very interesting because in all algorithms predicted that this ancestral sequence bind magnesium, and magnesium is the modern cofactor of this enzyme. We also did a structural alignment between the ancestral structure and the modern structure, and for our surprise, it's practically the same, the structure of the ancestral molecule and parts of the modern molecule. And in this picture, you can see that in A is the ancestral part of the RNA-dependent RNA polymerase, but derivated from the modern proteins. And in red, you can see the RNA-dependent RNA polymerase, but derivated from the translation of the tRNA ancestral sequence. And when you did the structural alignment, you can see that the structure is practically the same. Okay. We also did an structural tree, and here you can see an ancestral structure, and in here you can see the modern structure of different virus family. And this is very interesting because all of the ancestral structure are so close of this virus family, Leviviidae, Helviidae, Stoviridae, that are very ancient virus family. Okay, so in that moment, we had a suggestion that the first polymerase emerged by the translation of the tRNA ancestral molecules. This year, we published this article recently, 1 or 2 months ago, when we analyzed the origin of the DNA polymerases, and the strategy that we used was the constructed database with the modern DNA polymerases, reconstructed the ancestral structure, the ancestral sequence, and consequently the ancestral structure, and compare with the other polymerases. Here you can see the ancestral sequence to the family B of DNA polymerases, and you can find the exonuclease domain. That's a very important domain in the DNA polymerases. And you can also see here the DNA polymerase elongation subunit. This part of the protein replicated. This is the part, the catalytic part of the DNA polymerase. Okay, so this data indicated that the ancestral sequence had already had function of the function of replicated DNA molecules. But when we reconstructed the structure, the ancestral structure, and compare with the modern DNA polymerases and the heterotranscriptase, the ancestral structure is more similar to the heterotranscriptase than the DNA polymerases, the modern DNA polymerases. This is fascinating because when I looked for the sequence, I can identify elements of the DNA polymerases, but in structural terms, the structure is more similar to the heterotranscriptase. When we did a binding analysis of this ancestral structure to the DNA polymerase, we obtain a very interesting data. Here you can see in CM is the point of bind to magnesium, and in cyan here we had an RNA molecule. It's very interesting because the ancestral structure to the DNA polymerase had more affinity to RNA than to DNA. And again, when I look for the primary sequence, I can identify all elements of the modern DNA polymerase. But in structural terms, this sequence, this protein, this ancestral protein, is more similar with the heterotranscriptase than with the DNA polymerases. And we also did a structural tree, and for our surprise, here you can see the DNA polymerase ancestor. We had here the heterotranscriptase, and we had in the other branch here the family C, the DNA polymerase family C, family A, and family B, and the ancestral is structurally is more similar to the heterotranscriptase than all of the other families of the DNA polymerases. This data suggests that the DNA polymerase emerged from the heterotranscriptases. So let's talk about the DNA polymerases. We have today around 6 families of the DNA polymerases. 3 of these families are involved in the replication process, and the other 3 in the repair process. In bacteria, we had 2 families, the family C and the family A. And the family A also you can found in the mitochondria. This make more sense because everybody knows that the mitochondria, the origin of the mitochondria is from a bacterial lineage. In Archaea, in Eukarya, the main our DNA polymerase is of the family B. So this is very interesting because when you look the configuration, the distribution of these different kind of families of the DNA polymerases, clearly we can see a separated a separation between archaeal lineage and bacterial lineage. And when you compare the sequence of this family, we can find a similarity between them. So it's most probable that this different family had different origin. Okay, so to try to understand the natural history of the different kinds of families of the DNA polymerase, we published this paper at the beginning of this year. And we analyzed the different families, the distribution of the different families to try understand how the DNA appear in the different basal lineages. So now I will show some phylogenetic trees to different families, and you can see that you have a similar distribution when you look to the bacteria, and that is completely different when we look for the distribution, the phylogenetic distribution in Archaea and Eukarya. So the first tree is to the DNA polymerase, the family A DNA polymerase. Here, these three are rooted in the reverse transcriptase, because we have evidence that the DNA polymerase emerged from the heterotranscriptase. And the part that you observe here is that the first group to derivated here is compound. Just to remember, this is to the A family. The A family is found in the mitochondria and in the other bacteria. But in the other bacteria, in the regular bacteria, this polymerase participate of the maturation of the Okazaki fragments, not participation of the replication of the main genome. But in the mitochondria, yeah, this polymerase replicated the mitochondrial genome. So when you look at the distribution, the first group to the diversificated to the family A is compound only to DNA, family A DNA polymerase of the mitochondria. The second group to diversificated is compound only by viruses. And the last group to derivated is compounded by the other bacteria. Okay, so the structure to the origin and diversification of the family A DNA polymerases is and group of bacteria, the family emerge in a group of bacteria, pass this protein bilateral gene transfer to virus, and this virus is spread to the other bacteria in this clade. When you look to the when you look to the C family, we can just find in the bacteria, in the regular bacteria, we had a similar pattern. The first group to diversificated is compounded by a group of bacteria. In this case, this is very interesting because this group in your majority is compounded by extremophily groups. After this, the second group that diversificated is compounded only by viruses, and the last group is the rest of the bacteria. So the structure is the family C emerge in a group of bacteria, pass by lateral gene transfer to the virus, and virus is spread to the other bacteria. It's the same structure that you can observe to the family A and the family C. So this indicated that the basal lineage of bacteria had a genome of compounded by RNA and after the first diversification, these lineages independently acquired the DNA polymerases, and after this passed to virus, and virus spread it to these two families to the other bacteria. When you look to the polymerase, Archaea polymerase, that's the family B, we had other partner. Here again, these three are rooted in the reverse transcriptase. But here, the first group to diversificated is only compounded by viruses. And after this, we had an Archaea and Eukarya organisms. It's completely different. In this case, the data suggests that the DNA polymerase, the family B DNA polymerase, emerged in the virus lineage and passed to direct it to the basal lineage, Archaeal basal lineage. And the basal lineage of Archaea diversificated already with the DNA as the major informational molecule. So this data, those data are in line with the other data that you are present in a lot of time that suggests that we have not common cellular common ancestor. We had a community of the molecular biological system that encapsulated independently to formate the archaeal lineage and bacterial lineage, is still with our RNA genome. And independently in this two basal group emerged the DNA polymerases, twice in bacteria and once in Archaea. So the model that you are suggesting are present in this picture. We had a community that you call progenode. In that moment, we had only molecular biology, an open community of the molecular biology, molecular systems, and part of this biological system encapsulated to form the archaeal lineage. Part of this biological systems encapsulated to form the bacterial lineage. In both cases, the genome was composed by RNA, and to the archaeal lineage, this lineage received directly and structured family B DNA polymerases. In this case, the transition from the RNA genome to DNA genome occurred early in the archaeal lineage and later in the bacterial lineage, because in the bacterial lineage, diversified in other lineages, in other lineage primordial is still with the RNA genome, and independently development the DNA polymerase A in one lineage and C in the other lineage, and bilateral gene transfer to virus that pass this new polymerases, the families A and family C, pass to virus bilateral gene transfer, and virus spread these two new versions of the DNA polymerase to the other bacteria. So now I will summarize some ideas. The first informational molecule was RNAs. The RNA-dependent RNA polymerase originated from proto-tRNAs. The heterotranscriptase originated from RNA-dependent RNA polymerase. This is a suggestion because I'm working in this problem right now. I have not data that supported this information until this moment. And at this stage, the biological system had not yet compartmentalized. And the basal cellular lineage, Archaea and bacteria, had more than one origin. So we propose that Archaea, the cellular lineage, had two independent origins. And this, in this independent origin, the genome was composed by the RNA. The DNA polymerase originated from heterotranscriptase, and the DNA had at least three independent origins in cellular lineages. One to A family and other to the C family in the bacterial lineage, and one origin in the Archaea lineage. And these origins were completely independent. So I would like to thank Marco José, Thais Gaudencio, Francisco Prodosmi, and all group that work in this many works. Here's my contact. If you want to contact me to talk and to collaboration, please, will be a pleasure. Thank you so much. I open to question now.